Hey everyone, let's dive right into this explainer and look closely at the precise neurobiology of how our brains forge long-term memories. Have you ever wondered why you can easily recall the lyrics to some song you haven't heard in a decade, but you completely forget what you had for breakfast just 2 days ago? Well, today we're going to demystify a specific region of the brain called CA1. It's an area that literally acts as the gatekeeper of your mind. So how exactly does a fleeting moment become a memory that lasts a lifetime? To answer this, we're going to track a single piece of sensory information as it travels through the complex architecture of your brain to face its ultimate trial. By understanding this journey, we can unlock the biological secrets behind learning, retaining information, and how our actual life experiences physically shape our neural wiring.
Section 1. This seat of memory.
To find our gatekeeper, we've got to look deep inside the medial temporal lobe, right to the hippocampus. It gets its name from the Greek word for seahorse, just because of its kind of distinctive shape, and this structure is absolutely vital for memory consolidation and spatial navigation. Now, comparing the rodent and human brain reveals a really interesting divergence. Rodents actually have a larger hippocampus to cortex ratio than we do. Why? Well, because their survival relies heavily on complex spatial navigation, scurrying through mazes and tunnels. Humans, on the other hand, have a smaller ratio because we rely much more heavily on our massive cerebral cortex for higher executive functions. But despite these evolutionary differences, the fundamental, microscopic memory machinery within the hippocampus, it remains remarkably the same across species.
Moving on to Section 2. Journey through the hippocampus.
Think of it like navigating a complex network. Our sensory input actually has to follow a very specific sequential trail. The journey kicks off at the entorhinal cortex. You can think of this as the grand gathering place. It's the very first junction where all that sensory information from the cortex initially meets up before plunging deeper into the hippocampus itself. So the absolutely crucial point here is following this step-by-step path. First, from the entorhinal cortex, the signal travels via the perforant pathway to reach the granule cells of the dentate gyrus. Then, from there, it essentially rides the mossy fibers over to the CA3 field. And finally, the signal traverses this crucial axonal bridge called the Schaffer collaterals to reach the CA1 region. It's a highly distinct, sequential relay, and honestly, that's exactly why the hippocampus is such a preferred model for neuroscientists when they're studying memory. Section 3. CA1, the ultimate gatekeeper. All right, so our sensory signal has finally arrived at its final processing station, the CA1 region. Now, the mechanism CA1 uses to decide whether to toss the information or keep it forever is called long-term potentiation, or LTP for short. When neurons communicate via high-frequency stimulation, kind of like when you're intensely focusing on learning something completely new, the actual strength of the synaptic response increases. This potentiated synapse is a massive prerequisite for storing information. Basically, if the signal is strong enough, the CA1 gatekeeper grants its ultimate approval, locking that memory right in. Section 4. Strengthening the neural connection. Let's look at the molecular mechanics of CA1 synaptic plasticity. Imagine the CA3 neuron sending glutamate over to the CA1 neuron. When that high-frequency stimulation hits, bam, it kicks off our rapid chemical cascade. Calcium just floods into the CA1 neuron through these specialized NMDA receptors. And this influx of calcium, it acts as a super powerful trigger. It sets off cellular mechanisms that literally physically strengthen the synapse by adding brand new AMPA receptors to the membrane. This makes the whole connection far more robust for the future. Now, what's super fascinating here is that this process actually happens in distinct phases. We've got the early phase, which lasts about 1 to 3 hours. This is just a temporary chemical shift, and it doesn't even require any new proteins to be synthesized. But if that memory is going to be kept forever, it has to enter at the late phase, lasting anywhere from 2 to 24 hours. This is where the real heavy-duty structural remodeling occurs. It requires gene expression and entirely new proteins to physically build fresh synaptic connections. It's kind of like upgrading a dirt path into a concrete highway. Section 5, when CA1 goes offline. To truly appreciate the sheer power of this microscopic gatekeeper, scientists naturally had to ask, what happens if the whole system just breaks down? To find out, they utilized some pretty advanced genetic mice models. They actually engineered mice to specifically lack that crucial NMDA receptor, the one that lets the calcium in. But the catch is, they knocked it out only in the CA1 region, leaving the rest of the brain completely intact. When we compare a healthy control hippocampus to a genetically altered one, the visual evidence of this tweak is incredibly stark.
In a normal hippocampus, there's this distinct dark ribbon curving right through the tissue, which shows the presence of the NMDA receptors. But in a CA1 knockout hippocampus, that dark band is completely missing, specifically in the CA1 region. The gate has literally been chemically padlocked.
And wow, the behavioral results of missing this gatekeeper are just profound. In these knockout mice, long-term potentiation is completely prevented in the CA1 area. What's crazy, though, is that LTP remains totally normal in the earlier stages, like the dentate gyrus. But because that final CA1 approval is denied, the animal completely loses the ability to form long-term spatial memories. They fail dramatically in tasks like navigating a water maze. Even with a perfect starting path, without CA1, the memory simply evaporates. Poof, gone.
Section 6. Securing your lifelong memories. Zooming out from the cellular level of mice, we can actually see this exact same gatekeeper at work in some incredible feats of human memory. A classic example is the famous London taxi driver study from the early 90s. To get their license, these drivers have to memorize this sprawling, chaotic mental map of thousands of London streets. It's a grueling test known simply as the knowledge. Doing this requires extraordinary hippocampal memory consolidation. Just like a student pulling an all-nighter studying for a massive exam, these drivers are relying entirely on their CA1 gatekeepers. They're repeatedly firing, repeatedly authorizing that long-term potentiation to build up those robust spatial networks. Now, once the CA1 gatekeeper finally approves the memory, the sensory signal's journey in the hippocampus is technically complete, but its life as a memory has really just begun. From CA1, the newly minted long-term memory is transmitted down to the subiculum, and from there, it gets exported outward and safely distributed across the vast, complex networks of the cerebral cortex. That's where it'll be stored for years, decades, or, even an entire lifetime.
So as we wrap up, I really want to leave you with this one thought. Will the fleeting moments and biological concepts you just learned in this explainer pass through your own CA1 gatekeeper to become a permanent part of your mind tomorrow? It is absolutely fascinating to realize that right now, literally as you're listening to my voice, inside your own head, millions of microscopic gatekeepers are firing, chemically shifting, and actively deciding what pieces of today you actually get to keep forever. Thanks for joining me on this incredible journey through the mind.